Okay, well, my name is Omar. I'm in my final year of my PhD. My research is focused on solving some problems in the aquaculture, abalone aquaculture industry here in Australia. As a brief introduction, abalone is an edible snail. It's supposed to be delicatessen, but I think it's an acquired taste that you have to develop. Most of it, what is produced in Australia, is exported to Asia. And we usually don't find it in the markets, in the normal ones. They are in delicatessen stores, maybe at $90 or $100 a kilo. So as a student, it's out of my pocket. I only eat the samples that I get before putting chemicals and stuff like that. Okay, here I'm going to talk briefly about what I've been doing all this time. Okay, and I will start with the one in the left, which is the abalone gonad proteomic profile. And okay, this is the light. This goes. Okay. Okay, that's me trying to put together the proteins. Now, this is the Haliotis laevigata or green lip abalone in the left. And okay, a little bit of background. The abalone is mainly cultured in the coastal zone of South Australia, Victoria, Tasmania, and Western Australia. The main two species that are cultured is the black lip and green lip abalone, which is the Haliotis rubra and Haliotis laevigata. Well, it's a good commercial activity for Australia. In the years 2011 to 2012, well, the fisheries in aquaculture abalone, they made up to almost 200 million in that year. And on farms, they have a big issue because they are not able to control efficiently the spawning, which is the release of sexual gametes, either eggs or sperm. So these guys, they have invested a lot of time developing selective breeding programs to produce abalone that is resistant to diseases, that grows faster, that is efficient with the food. But at the time they want to cross these good organisms, either the male or the female, they don't spawn. So they are not making the most from the programs they develop. So we're trying to know what or how spawning is triggered in abalone. And here we're using proteomics based analysis. And the first thing that we wanted to do is to know, okay, what sort of proteins we have in the gonads of abalone. And having a look in the standard databases over there, well, about those are the numbers for proteins described in RevSeq and NCVI. And for green lip over there, there are only 30 reports. And well, Neil over here, and oyster, which is another commercially important species of mollusk. It has 12, 26. Okay, and the other issue is that the genome and the proteome of abalone is kind of nil. So the approach we took, well, the objectives for this project is to describe the protein composition of the gonad and provide a platform to enhance the abalone research by providing a database, protein database, and also to increase the annotated knowledge of abalone proteome in standard servers. So the approach we took, we sampled the gonad from the abalone, and then we did a normal protein, a total protein extraction, and put that into the mass spectrometer. And then to identify the proteins, we used three approaches. Two of them were to use the proteins available for mollusks in Uniprot and all the sequences from mollusks from NCBI. And the other approach, we gather all the information of cDNAs and ESTs and another sort of sequences that we found over there for other mollusks, and then we built a protein database. We translate that DNA information into a protein language, and we built another database, and we also search those results through this database. After that, well, we identified the proteins over there, and then this was a kind of a big task to remove the redundancy, to start with this one and checking that the hits we were getting, they were not already reported in NCBI or in UniProt. And then after that, we generate a final protein list. And the results we obtained from this approach, it was 122 proteins in female, 17 male, and 40 of those were common. And there were, from gene ontology, they were mainly involved in the structure and mainly in the structure. And then here we have a couple of pie charts to describe the functionality of these proteins. And well, in female, 44% of this was in ribosomal activity, and we associate that with the all the protein requirements during egg formation for the female. And on male, the majority of proteins were in energy metabolism because of we associate this as well with all the energy that is required by the sperm motility during gamete release. And well, in total, we identified 232 proteins. And by doing that customized database, we had a gain of over 60% of the proteins identified. The results, oh, sorry about that. From all those proteins we identify, we select and base our discussion in those proteins that are involved in one or another way with reproduction processes or with sexual maturation. And those proteins highlighted in green, they are proteins that are involved in the acrosomal reaction or fertilization, which in the case of abalone, it starts with the recognition between lysine, which is the majorly abundant protein in male, and well, in this case is vitellogenin, but the interaction occurs, the initiation of fertilization occurs with the interaction of vitelline envelope receptor for lysine and lysine. This work has been accepted for publication recently, and at the moment is in press in the Journal of Proteomics under the name of Exploring Genomic Data to Identify Proteins Involved in Abalone Reproduction. And then the other approach we are at the moment working on is to evaluate the abundance of proteins in gonads of abalone. But now we are comparing gonads from abalone that spawn, that failed to spawn, and after spawning, to see what proteins are differentially expressed in those tissues. And okay, well, I already said that. This is only to show that, well, we got the abalone from this time frame, post-spawning, and then after 12 hours that they were artificially induced to spawning. Those that didn't spawn, well, they were classified as failure to spawn. And what we did here, it was comparing failure to spawn versus post-spawning, spawning versus post-spawning, and all the combinations possible. We labeled the proteins using fluorescence. And then, well, we separate the proteins according to their isoelectrical point, and then in the second dimension according to the molecular weight. And then just in a laser scanner, we get the image to see what proteins were differentially expressed. And in this case, the red ones are down-regulated, and the green ones are proteins with higher expression. And those proteins with yellowish colors, they are supposed to not have any sort of differential expression. And I picked one of those, well, some of them, several, to sustain what I'm saying. And yeah, they are not, when they are comparing in their abundance, they don't seem to have any difference. Okay, and all the combinations were done. The protein patterns were generated for all the combinations possible. And after that, okay, this is a software that helps us to see how the proteins are being differentially expressed in one tissue versus the other. This is the same protein being differentially expressed in a spawning gonad, and this is a gonad that was failed to spawn, so there's a difference over there. Okay, after that, all those proteins were differential expression were selected, and they were analyzed again in the mass spectrometer. Then the identities were identified. Okay, I need to go further a little bit. Okay, from this work, these are the proteins that now we need to validate using MRM, which is a technique that allows you to quantify specific portions of a protein and, well, correlated in this case with the physiological status. And all these proteins were differentially expressed, but we only select those that we know they are involved in reproduction processes according to the literature. And, well, this is this shouldn't be, this is aquaculture actually. And we did the same for female and male, and at the moment I have a couple of manuscripts in preparation under those names, and also I'm putting together my thesis because I'm planning to submit in September. Well, this is quite advanced work already. And, well, our main conclusion is that this work is going to help, or it has the potential to assist the current methodologies that the Australian farms use to make abalone to spawn. And, well, I thank you to all our collaborators from UQ, CSIRO, supervisors, and the staff from Kangaroo Island, Abalone. My samples, they come from outside of the coast of Adelaide. And all these organizations, the Mexican Council of Science, Flinders, Kangaroo Island Abalone, the Australian CRC Seafood, and the Australian Abalone Association. And thanks to you for staying.